We're going to watch this little video about welcome Brookings APBIO students. In this slideshow we are going to look at chapter 17 and how is it that we can take information that's stored in the DNA and produce proteins in the cell that are going to provide all of the functions that cells need to keep the cell working. And we're going to start with the code molecule, our DNA. And that information is stored in our DNA, and that information is going to be passed through the mRNA to build proteins. Remember, proteins are the workhorses of the cell. They are going to provide all of the molecules that are going to be enzymes. They're structural. They can be transporters. They build muscle proteins. All of the things that we talked about when we talked about proteins in our molecule chapter are going to provide the molecules that actually function and do things in cells, and those cells ultimately obviously build our bodies. And so that is how DNA works. It works through proteins that have functions in cells. The central dogma, kind of the big idea about how information flows in cells, has to do with how do we connect the information that's stored in the DNA to turn that information into a final protein that's going to have function. So we're going to start with DNA in the cell, and DNA making a copy of itself during S phase is called replication. We're going to talk about that process a little bit and how it works. We're also going to be able to pass that information from our DNA molecule into an RNA form. That is called transcription. And then we're going to pass the information from RNA out to the ribosomes to make proteins. That process is called translation. And now the proteins are going to ultimately show a characteristic, a phenotype, as some sort of a trait that shows in the organism. And that is called the central dogma, that sequence of DNA to RNA to proteins, how the information passes. Metabolism has kind of taught us a lot about genes, and remember we talked about genetic disorders and looking at metabolic pathways that don't function correctly if something is missing, and help us to understand how genes work, and the idea that genes are coding for, a lot of times coding for enzymes that either function themselves or make other proteins or molecules that are going to function in the cell. So we looked at some different diseases caused by non-functioning genes. We looked at things like sickle cell. We looked at PKU, Tay-Sachs disease. And so lacking an enzyme is going to block a pathway of some sort and provide a method of seeing how that gene is going to work. So if we look at a normal, for example, a little biochemical pathway here, molecule A is going to be turned into molecule B by an enzyme, and that may turn into a second chemical reaction. Enzyme 2 can turn molecule B into molecule C, and C into D using a different enzyme, and so on. This is called a biochemical pathway. In any of those places along the way, if that enzyme is broken, if we have a mutation in the code that codes for that enzyme, it's going to block the pathway. And so that can lead to some diseases. We talked about some different genetic disorders that are caused by missing or non-functioning enzymes. If you think about a pathway, any of those steps along the way could be a place where the pathway is blocked, buildup can happen, and the molecules are not going to be changed into the next sequence in the pathway, and you can get some sort of a genetic disease. And so looking at pathways in the sequence and how they work is one of the ways that scientists have figured out how genes work and how the what the mechanism is for function in proteins in the cell. So we talked about things like Tay-Sachs, PKU. Those were all genetic disorders where we had an enzyme that didn't function the way it was supposed to function. And the answer to that question is yes. You are what proteins are what make you in a lot of different areas. The idea of one gene representing one enzyme was put out by two gentlemen, George Beadle and Edward Tatum, and they received the Nobel Prize for this idea that genes are coding for enzymes. They're coding for enzymes, and those enzymes are proteins that do chemical reactions and provide the molecules that are going to make the cell function. Their experiment that helped them to figure this idea out involved using yeast. Neurospora is a kind of yeast, and they were growing yeast in medium, in providing the food for the yeast to grow, and you can grow those spores by placing them in media. And if you provide them just minimal media, some salts, some sugars, they have the enzymes that are needed to take those building block molecules in the minimal media and convert them into the basic kinds of molecules, the vitamins and amino acids that the Neurospora need to grow. And what they did was they took spores and irradiated them with X-rays or ultraviolet light to cause mutations. And then they looked to see what kinds of growth they could get from those mutated spores. And if you grow them in media, complete media, that has all the building blocks, all the amino acids, all the vitamins, obviously those spores are going to be able to grow easily. That was their positive control. And then if they took some of those spores and they grew them in a minimal media without certain those building blocks, and basically just provided them with a few basic salts and sugars, and did not get control, they knew they had some sort of a mutant that was missing an enzyme that allowed it to make some of those vitamins or amino acids. And so what they did was plate out their mutants and then add back in, one at a time, add back in some of those basic building block molecules that the cell needed to grow. And if they were adding back something that the cell needed to grow that couldn't be made by that cell, they would get a negative reaction in their minimal media tube and a positive reaction. They could get the organism to grow if they provided the missing ingredient. And so they did experimental tubes with lots of different vitamins and amino acids, and they were looking for those mutants. By adding back one at a time and getting growth when that thing was added and no growth without that particular molecule, they could determine what the mutant was missing, the enzyme that was missing that produced that thing, whatever it was that was added back. So if you had no growth in minimal media but got those spores to grow when you added arginine, they could tell that the spores had a mutation in the enzyme that produced the arginine. And what they did was isolate a whole bunch of different mutants for different enzyme pathways and help them to come up with that idea that the pathways are being controlled by enzymes, and gene mutations, by knocking out those enzymes, they made the connection between genes coding for one gene, one enzyme. So we're going to take the information stored in the DNA, kind of like a blueprint for example, building a building, and transfer that information now into an RNA molecule, a message. It's kind of like making copies from that basic blueprint. The blueprint stays safe in the nucleus, and mRNA is our working copy that can go out to the cell. We can use that information, and always our blueprint stays in the nucleus. So that idea of taking DNA code and turning it into an RNA message is called transcription. That message now is going to go out to the cell, out to the ribosomes, and we're going to use ribosomes. That's kind of our factory that's going to use that blueprint coming from the RNA to build a protein by hooking amino acids together. And then that protein is going to do something in the cell. That process of going from message, RNA message, to protein is called translation. And that's what this chapter is about. How do I get the information stored in the nucleus out to the cell and build my proteins? And then proteins are going to function somehow in the cell to produce a phenotype, produce a trait, eye color, hair color, height, all of those things that make you, you. Okay, so when we're going from DNA to RNA, that process is called transcription. We're going from nucleic acid, DNA nucleic acid language to RNA nucleic acid language. Remember, DNA and RNA have a few differences. If we remember, RNA has ribose for its sugar component. The nitrogen bases, we're going to substitute uracil instead of thymine. Use bonding with A's now instead of T's. C's are still going to go with G's like we saw in our DNA molecule. Remember, RNA is single-stranded instead of double-stranded. And so we're going to take our information that's stored in our DNA and pass it into an RNA form. And there's lots of different kinds of RNAs. We learned about messenger RNA, ribosomal RNA, transfer RNA in bio. We're going to learn about some other kinds of RNAs also in this chapter, like siRNA and microRNAs. So the kind of RNAs that help with protein synthesis, taking the DNA code and turning it into a protein, are three kinds. First of all, ribosomal RNA, sometimes called rRNA, made in the nucleolus. Two subunits, a large and small subunit, make the ribosomes. And the RNAs, ribosomal RNA is going to combine with proteins now to form those ribosomes, form the ribosomes. And we talked about how bacterial ribosomes are different in size than eukaryotic ribosomes. We talked about the evidence for the endosymbiotic theory, and we talked about mitochondria and chloroplasts having smaller, more prokaryotic type ribosomes. If you think about the differences between bacterial ribosomes, prokaryotic ribosomes, and eukaryotes, that difference in size can be taken advantage of for some kinds of antibiotics. If we want to target and harm a bacteria growing inside our body, we can do that by targeting differences in their ribosomes so that the bacteria is harmed without harming the eukaryotic host. And we can base that because of the differences in the ribosome, sizes of ribosomes in bacteria and eukaryotic cells. Transfer RNA is the second kind of RNA important for protein synthesis. We have an anti-codon sequence on the transfer RNA that's going to match up with the codon on the RNA. There's a site to hold the amino acid that is going to be carried by the transfer RNA. And there are enzymes that load the amino acids onto the correct tRNA and use energy from ATP to do that. And the enzymes are called amino-acyl tRNA synthetases, and those enzymes are specific. Each enzyme, each amino acid has its own synthetase enzyme that's going to attach it to the correct tRNA as a carrier. The third kind of RNA that's going to help with protein synthesis is the messenger RNA, and that's going to actually be the message carrier. It's going to carry the information from the DNA code out to the ribosomes. So if we look at this process now, how do I make an mRNA from a piece of DNA? We're going to use the DNA strand as a template, just like we did when we did making our copy. The untranscribed, the strand that we don't use to read from, is called the coding strand, because it has the same sequence as the RNA, except for we're going to substitute T's for U's. The template strand is the strand that's going to be transcribed. And now we're going to open our DNA and use our strand as a template now. And there's a little kind of a bubble, just like we opened for a replication fork, that site of where we're doing the replication. Now we're going to open the DNA and have a little bubble where we're going to be copying off of the DNA to make a piece of RNA. And the enzyme that's going to do this is RNA polymerase. Just like we had DNA polymerase to make our copy of our DNA, RNA polymerase now is going to use the strand of DNA as a template to make our little piece of RNA message.
Transcription is the first step in the expression of a gene. It is the formation of a specific RNA sequence from a specific DNA sequence. Transcription initiation occurs at a promoter, a region on the DNA, where RNA polymerase binds. RNA polymerase attaches to the promoter and begins to unwind the DNA. At the initiation site, the polymerase begins reading the DNA template strand and building a complementary RNA strand from free nucleoside triphosphates. The RNA strand grows by the addition of these substrate molecules to its 3' end. This is the elongation phase of transcription. Note that the bases in the nucleotides of the RNA strand form pairs with the bases in the DNA strand. Also, note that the RNA nucleotides contain the sugar ribose, which has a 2' hydroxyl group, whereas the DNA nucleotides contain the sugar deoxyribose, which lacks this hydroxyl group. RNA polymerase functions similarly to DNA polymerase. It adds nucleotides to the 3' hydroxyl group of the last nucleotide. During the reaction, a pyrophosphate ion is released and broken down, releasing energy that fuels transcription. The DNA double helix rewinds as the RNA polymerase moves through. Like the unwinding, the rewinding is an energy-requiring process that is accomplished by RNA polymerase and fueled by the breakdown of pyrophosphate ions. When RNA polymerase reaches the termination site, the RNA transcript is released from the template. The DNA rewinds completely, and the RNA polymerase dissociates from it. The DNA and RNA polymerase can then participate in other rounds of transcription.
Okay, so let's go back to our slideshow. And we talked about this process now of using the DNA template strand to add in the matching complementary RNA little subunits and build a strand of messenger RNA. And we're going to build in the 5' to 3' direction, just like we did when we built our DNA copy off of our DNA strand during replication. We're adding to that 3' end again. And let's look now at what's going to be involved here. We have 3 kinds of RNA polymerase. Now remember, that's the enzyme that is reading the DNA and building the complementary RNA message. RNA polymerase 1 is used to make ribosomal RNA. RNA polymerase 2 is transcribing genes into messenger RNA. And RNA polymerase 3 is going to transcribe the code to make the tRNA molecules. Each of these has a specific promoter sequence at the beginning that it recognizes to start reading the information, and RNA polymerase then, those different forms are going to make the different forms of RNA from the DNA code. How does the cell know which gene is going to be read? It looks for those promoter regions. Remember, promoter region is where the polymerase, the RNA polymerase, is going to bind. It's the binding site at the beginning of the gene. There's a little promoter sequence. Tata boxes, we talked about Tata boxes in bio, sequences with many AT repeats. Remember, the bond between the A's and T's is a 2 hydrogen bond compared to GC's having 3 hydrogen bonds. So it makes this area a little easier to open up and get going on reading the information. Tata boxes are part of that promoter sequence. The binding site now for the RNA polymerase, we have transcription factors, different kinds of proteins that are involved in helping this process along. And also we have enhancer regions farther up the strand, a little farther down, that are going to help to encourage binding of that polymerase by folding in different 3D shapes to help that process happen. And all of those are going to signal the polymerase enzyme where it is supposed to start transcribing and reading the information. Transcription factors are involved. There's a number of proteins, initiation complexes that help to bind to that promoter region and get the polymerase going. Sometimes these can be hormones. Remember, we talked about the way that steroid hormones work by getting through the plasma membrane, binding to receptors. When we talked about cell signaling, binding to receptors in the cytoplasm of the cell, passing through the nuclear envelope and actually attaching to the DNA to turn on and stimulate the production of turn on different genes. And so all of those can be important factors that help the transcription process, turning on and off different genes. They trigger the binding and help position the RNA polymerase to get it going to read the message. So we're going to match our bases now, complementary, matching RNAs to the DNA strand. A's with A matches with T in the code. U, remember we can't put T's in message. If I'm building RNA, U is going to match with this A, C with the G, A with the T, and so on. And we're going to build our code now by joining those nucleoside triphosphate subunits into the strand. Once we have our DNA transcribed into message, in eukaryotes, if you look at how the gene is laid out, the gene sequence is actually not continuous. There are little pieces that need to be removed from the final piece of RNA, and exons are the one part of the code that's going to be expressed in the final mRNA message. The introns are what we call the junk DNA that is going to be cut out. Once the message is produced from the DNA sequence, it's going to be edited before it's used by the ribosomes, and that editing process is going to happen by removing these in-between sequences and putting together the pieces that are going to make the final part of the message. Introns are cut out, exons are expressed. So if you look at the code now, here's my piece of DNA that contains those introns and exons. We're going to copy it directly off of the DNA. So we make what's called a pre-mRNA molecule, a pre-transcript. And that little piece of message now is going to have parts of it removed. We're going to take out those intron pieces, and they're going to be recycled back. They can be recycled back to build new message. And the exons are expressed. Those parts of the code are going to be strung together to make a final piece of RNA that's going to be read by the ribosomes. So how does the cell do this? How do we do the splicing? The post-transcriptional processing, that means processing after we've transcribed into an RNA. We're going to take that primary transcript and we're going to do something called splicing, editing out those intron pieces and putting together the exons that are left over. We're going to make what we call a mature mRNA transcript. So this is only in eukaryotic DNA now, not in prokaryotes. Prokaryotes do not edit out their introns. They do complete sequence directly off the DNA, and there is not editing to that message like we see in eukaryotes. So the primary transcript that comes off the DNA is going to be edited to make a mature final piece of mRNA that's going to go out to the ribosome. So let's look at what's this process involved. The idea of introns and exons and that DNA is edited was discovered by these guys. They got the Nobel Prize in 1993 for discovering this process of editing our message to remove those introns. And if you think about it now, splicing has to be pretty accurate. If we snip out more than we need or make the cut in the wrong places, it's going to knock off the reading frame in that message code. And remember, reading frame mutations can cause serious problems if putting the amino acids together correctly, if the code is off by one place. So remember, we're going to read in little groups of threes. So if this is my original message, this is my edited message now that's going to be modified by removing those introns. And now I'm going to take this final transcript and I'm going to read that in little groups of 3 to produce a protein. Each little code is going to stand for an amino acid. So if I edit incorrectly, even just getting off by one letter, that can cause a reading frame shift, and it causes the protein that's made to be made incorrectly, especially if we put a stop codon in the middle of the message. It stops the reading frame there. So the enzymes that do this splicing have to be very specific and very careful about where they're cutting. And the enzymes that do this contain molecules called SNRPs, small nuclear RNAs. That's another kind of RNA. Remember, we talked about there are other kinds of RNAs besides message, ribosomal, and transfer RNAs. Small nuclear RNAs combine with proteins to make editing complexes. And their job is to attach to the message and recognize the splice sites where the pieces need to be cut out. They fold the RNA message and put the exon ends together and splice out those little introns out of the middle. They go together to make big splicing complexes called spliceosomes. And it involves several what we call SNRPs, small nuclear ribosomal or small nuclear RNAs, and they are going to be the enzymes that recognize those splice sites and snip out the introns. They're called spliceosomes. And they are going to cut and paste now to remove those intron pieces. So think about that. You have RNAs that are acting as enzymes. RNAs acting as enzymes. Remember when we talked about enzymes, and the majority of enzymes in biological systems are proteins. Proteins act as enzymes. And we talked about some different kinds of proteins that are enzymes. But these are an example of RNAs that are acting as enzymes. We call them ribozymes, RNA molecules that function as enzymes. And in some organisms, those pre-RNA little sections are actually edited, self-edited, and they cut out their own introns. And it's the SNRPs, it's those ribosomal, those small nuclear ribosomal RNAs that are cutting out and acting as enzymes. So we're going to watch a little video here about the processing and what happens to the message after it's been made, and take a look at how it's different in prokaryotes and eukaryotes.
Gene information is processed differently in prokaryotes and eukaryotes. Because prokaryotes lack a nuclear envelope, messenger RNA, mRNA, can associate with ribosomes in the cytoplasm as the mRNA is being formed. Translation of mRNA into protein begins before transcription is complete. Individual bacterial mRNA molecules often contain transcripts of several genes. By placing genes with related functions on the same mRNA, bacteria coordinate the synthesis of these proteins. These clustered genes are referred to as an operon. Because eukaryotic cells possess a nucleus, their mRNA must be completely formed and must pass across the nuclear envelope before translation. Eukaryotic mRNAs are modified before they are translated. Introns are removed and the remaining exons spliced together. A 5' cap and a 3' poly A tail are added. The processed mRNA travels to the cytoplasm where translation occurs. In contrast to prokaryotic mRNA, eukaryotic mRNA usually specifies only a single protein.
Okay, let's take a little piece at the spliceosome process.
Most eukaryotic genes are composed of numerous short coding sequences called exons, embedded within stretches of non-coding sequences called introns. The initial messenger RNA molecule, or primary transcript copied from a gene by RNA polymerase, is a faithful copy of the entire gene, including introns as well as exons. Before the primary transcript is translated, the introns are removed by a process called RNA processing, or RNA splicing. Particles composed of proteins and a special type of RNA called small nuclear RNA, or snRNA, play a role in RNA splicing. One kind of small nuclear ribonucleoprotein, snRNP, contains snRNA that can bind to the 5' end of an intron by forming base pairs with complementary sequences on the intron. A different snRNP binds to the 3' end of the intron. Additional introns interact, causing the intron to form a loop, thereby bringing the two ends of the intron together. The large complex of snRNPs, called a spliceosome, then excises the intron and the exons are joined together. The snRNPs are then released.
Okay, let's go back to our slideshow. So we're going to use those spliceosomes to remove our introns and process the message into a final form. What this process allows the cell to do is to edit DNA, the same piece of DNA, in different ways by putting together different pieces of that transcript and splicing out different pieces of what would be considered the introns to actually use the same piece of DNA to make different messages. And so if you think about that process, what it allows the cell to do is use that same sequence of DNA in a variety of ways. And so if you think about it now, it's starting to get a little fuzzy about what we're calling what is a gene, because if we can use that same piece of DNA to make different proteins, to make different messages, it kind of starts to make us modify our one gene, one enzyme theory a little bit. Once the mRNA is processed, is edited by removing introns, it needs to be have a little more final processing before it leaves the nucleus. And so there is a cap, a GTP cap that's added at the 5' end. And there's a poly A tail that's added on the 3' end, multiple AAAAs added here. And it is going to be a protective cap, a protective tips on the ends of the message that prevents it from being broken down by molecules in the cytoplasm that are going to destroy those mRNAs. It protects the molecule and also helps to get it out through the nucleus. So we're going to add a GTP cap to our 5' end, and we're going to add a poly A tail to our 3' end that helps that mRNA have a longer life once it gets out to the cytoplasm. So we're going to take our DNA code in the nucleus, we're going to pass that information, change it into an RNA message. That process is going to be transcription. And now we're going to take that message out to the ribosomes out in the cell and use it to make proteins. Ribosomes are a protein factory. And those proteins then are going to go on to do something in the cell. And so we're going to stop right here. There's another slideshow video coming that you should also watch after this one that has to do with that last process of, now that I've made my RNA message, how do I turn that into a protein? So stay tuned.